So serotonin is a very complicated transmitter system with a great many potential postsynaptic and presynaptic receptor targets. But this is, an illustration up here of, again, a synapse. Let's say this is now our serotonergic neuron, a neuron that's, primarily releasing serotonin at the synaptic, connections here at the axon terminals. And so if we look at an axon terminal for serotonin, you could see what's illustrated here is the amino acid tryptophan, which is the widely available raw material for the manufacture or synthesis of serotonin. There's actually two enzymatic steps, right, that take tryptophan through 5-HTP down to 5-HT or 5-hydroxytryptamine, which is serotonin. Then, of course, there has to be a vesicular transporter that allows for the entry or the packaging, right, of the 5-HT or the serotonin inside the vesicles at the axon terminal. And remember, this is the same monoamine vesicular transporter that will also transport dopamine and norepinephrine, our two catecholamine neurotransmitters, and pack those inside, vesicles in dopaminergic and noradrenergic neurons. So serotonergic neurons also express the same gene to produce the same, vesicular monoamine transporter to package, fight those concentration gradient forces, package that serotonin inside vesicles. Then, of course, there is, the arrival of an action potential and release of serotonin into the cleft, right? Fusion, exocytosis, that whole docking complex aspect. And then there is, again, a selective presynaptic reuptake transporter for serotonin. So this is another special gene that's going to be expressed, just in these serotonergic neurons that's going to actually, transcribe and be translated into a transporter that utilizes ATP, right, to grab serotonin, this time out of the synaptic cleft after it's been released, and, force it back inside the axon terminal. That's a selective, reuptake transporter for serotonin. And then once back inside the cleft, again, remember for the monoamines, dopamine, norepinephrine, and serotonin, there is MAO, which is monoamine oxidase, which is an enzyme that will break down, any of the monoamines. In this case, in the serotonergic axon terminal, it'll break down serotonin. Now, postsynaptically and also to some extent presynaptically, because there are autoreceptors, there are 15 different subtypes of serotonergic receptor. A majority of those, 14 of them, are actually metabotropic, coupled to different G proteins, while only one of them is ionotropic. So you can imagine this is an extraordinarily complicated, set of receptor targets for this neurotransmitter, which is, of course, very much involved in psychological health and mood states.